The context of the research that gets carried out in my group, which is a biophysics group in the Department of Mechanical Engineering at UCL, is tissue engineering and its wider implications. I mean, everybody has heard of electrosprays or electrospinning, and it has been carried out. These processes have been carried out for the last 100 years, if not longer. But what we've done that is different and that is new is that we've used these electric field driven technologies to actually process living cells. We were the first group to do this, and we found that obviously people would say, well, and people did say, that cells do die when you apply a voltage to them. But what they didn't know was that cells don't die, because although the voltage was in the several thousands of volts, the current is in the nanoamperes, which is the opposite to electroporation, which is where you use 230 volts with 55 milliamperes or whatever, and then get the cell membrane permeable, and you get your genetic construct in. And during that process, you lose about 70% of cells, depending on cell type. But our process does not do that, because the current is very low, and the immediate application where such a technology has been used is where taser guns. So you can stun whoever you want to stun at with 15,000 volts, but the current is very, very low. But you don't kill them. So in the context of our work is where we are using electrosprays and electrospinning, which now referred to as bio-electrosprays and cell electrospinning, to actually process cells as either beads or as fibers to make tissues. And tissues that could be used for repairing, replacing and rejuvenating damaged or aging tissues. The methodologies that we used, as I mentioned before, electrosprays and electrospinning, have been known for several years. What to the layperson, these are technologies that uses an electric field or a potential difference between two electrodes, and you can either spray out these cells as droplets containing cells, or pull them out in the form of a filament, a continuous filament of cells within fibers. Obviously, when you collect these using certain chemicals that are biologically compatible, you can cross-link them and make cell-containing beads, which could be used for drug delivery, drug discovery, and a whole plethora of applications to building tissues, much like scaffolds, where these continuous fibers are collected over a function of time and make, if you like, a piece of shirt or something as even thick as a mattress. And let me also say at this time that these are the only processes that are completely biologically fully tested, both in vivo and in vitro, and have been shown to have no harm on the cells. We've shown that these processing approaches do not harm cells, ranging from primary cells immortalized to stem cells, including iPS, to even whole fertilized embryos, showing that the electric field that is used doesn't affect their embryological development in the case of early fertilized embryos, to various primary and stem cell types that could be moved from a specialized to from a unspecialized to a specialized lineage. So, and also in terms of their function and their ability to behave as expected in comparison to controls. So that has been our main piece of work that we've been concentrating on. But now what we are hoping to do is to take that to the next phase, is to prolong our preclinical studies to larger animals, so as to make, if you like, tissue constructs that will be used for repairing. Hearsay, if you like, or a lot of things being said that these technologies could build an entire organ. I personally think that is actually complete Star Trek or fiction. However, I think in the next 5 to 10 years, using our technologies, I think we will be able to patch an entire organ. And therefore, although that may be also quite a huge feature, that is where we'll be at in the next 5 to 10 years. But replacing an entire organ is completely science fiction at this point in time, in my opinion.